Hello everyone, welcome to my YouTube channel. Today we are going to discuss about how nanotechnology is having differences in length scales, or how we can see the materials at how we can compare the normal existing materials with the nanoscale. So in introduction, might have explained in one slide. So here we are having a separate topic that we will discuss. Very small topics. So nanotechnology has a great impact on over every field of science and technology, as materials are important, because without materials there is no field, right? So nanotechnology has a great impact on every field of science and technology. Why? As materials gain extraordinary properties when reduced from bulk to the nanoscale. Whatever the material you take from the bulk, and if you are reducing it to the nanoscale, then you are going to get some extraordinary properties. So that's why the nanotechnology plays a very important role, or has a great impact, okay, on every field of science. So bulk materials, when reduced to less than 200 nanometer, that it can be 1 to 200 nanometer, okay, material can be from 1 nanometer or it can goes to up to 200 nanometer. They do exhibit different properties, especially when reduced to less than 100 nanometer. So which falls between, this particular scale is there, 1 to 200 nanometer. This actually falls between the molecular size and to the atomic scale. So at this scale the properties changes, which makes nanomaterials very special, and we have seen how does the property changes. Okay, so the property changes based on we discussed about the gold nanoparticles, which we are changing its melting point, which are changing its as we are changing the the size, its optical properties were getting changed. Okay, so all these things will come into picture. So where we can say that certain things are changing. So this whatever the thing, the properties, not maybe say for example, it can be the optical property, it can be the mechanical property, it can be the electronic property. All these things can change. So that's what makes them extraordinary. So what I am getting the conduction at the bulk scale, so I'll be getting definitely the better than the better in the nanostructures. So we are having, this is how the scale we can compare. We do have the natural things at macro scale to the micro to the and nano. And similarly we are going to compare the man-made things which started from the macro scale, like millimeter to the micrometer to the nanometer. So all of you know the scales. This is the scale what you can give, that is from 1 centimeter to the 0.1 nanometer. So this is the scale length, and this is how the things goes. We will come from the bigger aspects, like from centimeter. 1 centimeter we are having around less than 1 centimeter we are having around 10 millimeter. So in natural things we are having the dust mite. So dust mite, whatever the thing will be there, that will be around 200 micrometer. Or if you still want still bigger size, we are having ant. So ant is of around 5 millimeter. So you might have seen how tiny it looks. So that's the just beginning of comparison here. And the man-made things we are having as tiny as ant, we are having the head of the pin. Okay, so head of the pin is almost around the 1 to 2 millimeter, and then you are going to see about dust mite. Dust mite will be around 200 micrometer, whereas we are having micro electromechanical system with compared to the man-made things. Okay, compared to the natural things, we are having around 10 to 100 micrometer. So here you can we have got a lot of examples in my natural things. We are having the human hair, which is having 60 to 120 micrometer wide, whereas we are having the fly ash, okay, that is also around 10 to 20 micrometer. So these all things you can comprise into the micro electromechanical systems, which are prepared by the engineers. And we do have the RBCs. We know that in our blood there are three things: RBC, WBC, and platelets. So every particles or every molecules inside our body has got their wonderful functions. They are truly the natural nanostructures which are performing a lot of work inside our body. For example, RBC. In RBC, we are having the red blood cells. That means in this particular red blood cells is responsible to carry oxygen, okay, throughout all our body. So once we just take the oxygen, when we inhale oxygen, it will go to lungs, and from the lungs there will be exchange mechanism to the muscles. So the oxygen will go to the muscles, okay, and from there it enters to the, now different parts of the muscles it has to go, right? So then it enters to the bloodstream. So once it enters to the bloodstream, then the heart will keep on pumping with the help of lung. So that's how the blood pumping takes place, and this pump blood will go to all parts of our body, and that's where all parts of our tissues are getting oxygen. Imagine if the oxygen is stopped, that means almost, if there is no oxygen, obviously the person will die, right? So imagine if the RBC is not working properly, and inside RBC there is some agent is there which takes this oxygen to every part of our body, that is hemoglobin. Okay, that's the protein which holds the oxygen and which binds with oxygen, and it takes oxygen to all parts of which carries oxygen through the bloodstream, that is in RBC, in the blood. That's how. Without oxygen, obviously, definitely the person is going to die. Why? Because in order to the muscles are there, the organs are there, the brain is there, the heart is there, but if oxygen is not there, nothing will work, because oxygen has to be carried out. And that's see how important, and why I am taking all these things are, see how important these scales even nature is teaching us, or we all got inspiration from all these kind of molecules. Then only we are able to create MEMS or NEMS, nano electromechanical systems, or micro electromechanical systems. Whatever we are having microelectronics, they have to give the big credit to the naturally existing things which are at the microns scale, and which are doing a lot of the engineering work which cannot be done by the human made things. Even though we say that we have done a lot of significant advancement in the field of science and technology, but yet we cannot copy a simple RBC's function yet. See, that's how, because they are naturally engineered, right? The naturally engineered protocols, all those things are from DNA to the protein to the molecule, all these molecules which they are doing excellent job in our body, right? If I'm seeing, if I'm talking, there are n number of proteins which are involved, n number of the vitamins which are involved, n number of cells which are involved. Okay, all these things are functioning. That's why I'm able to talk, or that's why I'm able to walk, or whatever it is. So that's how things goes. Even as you go on reducing the size, the applications are more and more impact they are having. That's what this particular topic is trying to explain you. So next we are having the comparison. Next comparison, we are having the zone plate X-ray lens, which is prepared in man-made things. For that, around, again, it is almost nearly around 35 nanometer. The outer space from one space to another space, that spacing from one ring to another ring will be around nearly 35 nanometer. So that if you are going to compare that with any kind of molecules which are naturally available, we are having ATP synthase. Okay, those who have studied, have taken the biology in your PUC will be knowing what is ATP, right? Adenosine triphosphate. Okay, so this synthase enzyme will have the diameter of 10 nanometer. Okay, so this is actually the three-dimensional structure of ATP, which is with the help of bioinformatics tools and techniques that has been engineered, and nowadays it's available in NCBI, that is National Center for Biotechnology Information, right? So next we are talking about the man-made things where self-assembled nature-inspired structures are there. Many tens of nanometer, maybe it's 100 or 200, 150 nanometer, like that. Next we are having the nanotube electrode, which is having, see, imagine this scale is 1 micrometer. See here, all of you. So here this scale is 1 micrometer. So now you can just simply place this scale here. So this will be just 1 micrometer. Okay, so these are what different what you say, the products, the components inside the electrode. So nowadays we are having different components in the given electronic circuit or something like that. So now we can join these things with the help of, can you see one line here, the single line. So that single line is a single carbon nanotube which is making connection between these two and these two, right, the white line. So that white line is having around the diameter of 1.3 nanometer. Can you just imagine this particular small things? Okay, as 1.3 nanometer. This we were able to see only under the electron microscope. With the help of electron microscope only we can see that. Without electron microscope we cannot see this. We can't even imagine to work at this thing. So all credit goes to the IBM, which developed scanning electron microscope, atomic force microscopes, scanning tunneling microscopes. With the help of all those things only we are able to carry the Moore's law from macro to the micro to the nanoscale. So that's how these carbon nanotubes are nothing but it is prepared by the graphene sheet. The graphene sheet is again prepared by the graphite. We know, all of you know that the graphite is a three-dimensional structure. So it is made up of the different small layers, and those small layers are not a single layer. If you are taking, that is graphene, and graphene, its diameter will be, its thickness will be in nanoscale. So that's why it is also comes under nanomaterial. So two-dimensional nano sheet. So if you are folding this nano sheet, you are going to get the carbon nanotube. So if you are folding it in horizontally, you will get the zigzag structure, and your structure will be called as zigzag carbon nanotube. If it is folded with the horizontal, then we are having the armchair. This is what the armchair. See here. So it looks like this, like this, like this, and like this. That's why it looks like an arm of the chair. So that's why it is called as armchair. You might have studied that. Next we are having the chiral. So chiral is nothing but, if while folding this sheet, if I am making it as a cross with some angle, okay, so then you will get that particular chirality. So in chiral we might get as a electronic property as an insulator, armchair we might get as a conductor, and in zigzag we will get as a semiconductor. So this is how the carbon, one single material only we can exploit, and we can get all three electronic properties. Isn't that cool? So with the help of the nature inspired things, we were able to create all these things. That's how the scale can be differed. So now speaking about this 1.3 nanometer, that we can compare with the diameter of the DNA. Everybody know that we all are here just because of DNA, right? So DNA's diameter is around 2.5 nanometer. So next we are coming to the man-made things which can be comparable with the atoms. So here atoms of silicon, that is around 0.075 nanometer, not even 0.1 nanometer, lesser than that, 0.078 nanometer. So not the silicon, but see the space between one single atom. You can see the silicon atom. So that's the beauty of electron microscopes. We can even see the atoms. So with the help of this thing, the comparison by inspired this thing, nature, we were able to prepare the quantum coral of 48 iron atoms, okay, on a copper surface. We have a copper on the surface, 40 different quantum coral atoms, iron atoms have been placed, okay, positioned, okay, one at the time with the STM tip. This is scanning tunneling microscope, which is designed by developed and invented by the IBM scientists who got Nobel Prize for this. Okay, why? Because why so it is important? There are all electronic microscope, and a number of electron microscopes are there, but nothing can beat scanning tunneling microscope, because the scanning tunneling microscope works based on the tunneling mechanism. Tunneling mechanism is something, it's a concept of quantum mechanics. In normal physics you might have, in physics we have studied. So here the electron microscope tip will be there, no? So that will be fixed with one atom, single atom. Okay, just imagine, single atom will be fixed on the tip, and when it is scanned on the surface of any sample, the atoms which are there in the surface, I can pick single atom from the given sample. So with the help of the similar STM, same STM, they were able to pick single atom of iron, and they were able to prepare quantum coral. of with the 48 different carbon iron atoms. Initially they made, when they discovered STM, they made every single atom, they made with the help of single atom, they created the IBM logo. That's what it's like. Around almost we are having the whole diameter of this is 14 nanometer, the whole diameter. That means iron 48 atoms are there, right? So whole diameter will be around 14 nanometer. So every, imagine, divided by 48 divided by almost 14, right? So here atoms spacing is 0.078 nanometer. So that's what the nature has got this scale, and all these are significant. And when you are reducing from the head of the pin to the such a level, we are having significant application from the MEMS, from the zone plate X-ray lens. Then we are having self-assembled nature-inspired nanostructures, nanotube electrode, and which has got a magnificent applications in most of the electronics field. Then we are having the quantum corolla of 48 atoms. So that's how the things works. So we are having the nature inspired things at different scales. That's why we can call it as a length scale. Okay, under this scale you can explain all these things. So next we are having the nano gold. I can already discuss the same thing. You can also copy paste. So while the strange things happens at the scale, nano scale or small scale, if we keep cutting until the gold pieces are in the nanoscale range, they don't look gold anymore, they look red in color. So 12 nanometer gold particles look red in color. In fact, depending on the size, they can turn red, blue, yellow and other colors also. See, I am just comparing here and there, then we are getting this particular different colors. We are having the red, purple, all these things. Why? Different thickness of the materials reflect and absorb light differently. That's why the particle changes, the color changes, the optical property changes. At 50 nanometer something is there, at 80 nanometer something else is there. This is what we discussed in the earlier classes, and same thing can be written for this topic also. Why is nanotechnology so cool? In that we have studied. So here imagine at bulk we are having the color as a gold, melting point as 1064 degrees Celsius, color will be the gold. So whereas here if I reduce that into the 100 nanometer, I am getting the melting point with almost similar, only 64 degrees changes, but lambda max around 575 nanometer. Okay, and color is purple pink. Lambda max is nothing but the wavelength, the maximum wavelength at the more absorption takes place. Okay, the wavelength will, the color is purple pink. If I reduce that to the 100 to the 20 nanometer, I am getting red in color. If I reduce that into the 1 nanometer, I am again getting brown yellowish color. Okay, there is a lot of difference, right? As the size changes from 1 to 20 nanometer, the properties are changing. From 20 to 100, again the properties are keep changing. So that's the beauty of the scale or a length scale at the nano structure. I hope you understood. If any doubts are there, please write it to me, email. Okay, ID is already there in the given playlist. You can write it. Next topic we are having the variations of physical properties from bulk to the thin films and then to the nanomaterial, that we will discuss in the next class.